हे गाइस, क्या हालचाल। नया दिन नई शुरुआत। सो व्हाट्स इन अ लैब। जी हाँ, आज का हमारा टॉपिक है एक लैब में आखिर होता क्या है। डिफरेंट इक्विपमेंट्स, टेक्निक्स, इंस्ट्रूमेंट्स, सॉफ्टवेयर्स एंड डिवाइसेस। सो मैंने आज फार्माकोलॉजी, फार्माकोग्नोसी, फार्मास्यूटिक्स, फार्मास्यूटिकल एनालिसिस, फार्माको इन्फॉर्मेटिक्स, बायोटेक्नोलॉजी, इन लैब्स के बारे में थोड़ा सा डिस्कस करने वाले हैं। ऑफ कोर्स फार्माकोलॉजी में प्रीवियसली ऑर्गन बाथ वगैरह काफी ज्यादा यूज होता था। स्टूडेंट्स ऑर्गन बाथ, काइमोग्राफ वगैरह जो बनता था। फॉर न्यूरोलॉजिकल डिजीज वी हैव मॉरिस वाटर, रोटा रॉड, एडीज हॉट प्लेट, अनाल्जेसियोमीटर, एक्टोफोटोमीटर, पोल क्लाइन टेस्ट, कन्वर्सोमीटर्स, सिलेंडर टेस्ट। काफी ज्यादा टेस्ट होती है जो कि आप न्यूरोलॉजिकल बिहेवियरल टेस्ट, फंक्शनल टेस्ट वगैरह भी के लिए काफी ज्यादा केमिकल्स अगर हम बात करें तो वो बायोकेमिकल्स, इन्फ्लामेटरी एसिड्स, अलाइजा, वो अलग होता है। यू हैव लैंगेनडॉर्फ, यू हैव लॉट ऑफ इंस्ट्रूमेंट्स दैट कैन बी यूज्ड इन फार्माकोलॉजी लैब। इसका डिटेल वीडियो भी मैंने एक बनाया फार्माकोलॉजी के लैब का, हमारे इसी चैनल पे आप देख सकते हैं। सॉफ्टवेयर ऑफ कोर्स आप ग्राफ पैड, प्रिज्म यूज कर सकते हैं, आप सिग्मा स्टैट यूज कर सकते हैं, बायोपैक। ये सारे सॉफ्टवेयर आप स्टैटिस्टिकल एनालिसिस के लिए, इफ यू वांट टू आइडेंटिफाई विद इन ग्रुप्स कितना स्टैटिस्टिकल डिफरेंस है, इंटर ग्रुप एंड इंट्रा ग्रुप वेरिएशंस, क्योंकि एक ग्रुप में 6 एनिमल्स है, उसके अंदर भी वेरिएशन होगा। 2 अलग अलग ग्रुप्स होंगे कंट्रोल डिजीज, बट उसी कंट्रोल में और भी सब क्लासिफिकेशन होगा। So for that, ANOVA test, t-test, post-hoc analysis like Bonferroni's test, Tucky's t-test, you know, honestly significant difference test, HSD. So काफी सारे टेस्ट है जो आप post-hoc कर सकते हैं। Techniques, definitely you can perform a bioassay, you can perform pharmacokinetics characterization, जैसे plasma level concentration, urine concentration of drug, preclinical behavior like the safety pharmacological studies and toxicological assessment also you can do. Organ specific studies, liver, lung, kidney, intestine, heart, skin. There are different systems in the body: cardiovascular, central nervous system, endocrine system, lymphatic, autonomic, dermal. You can work on any system that you wish to work for. Skeletal-muscular system, lot of systems there. Instruments, microscope, definitely if you perform histopathology, we need a microscope to identify. Camera lucida sometimes is required. Many a times, in pharmacognosy, when I move to the pharmacognosy also, you need a microscope, definitely, because microscopic characteristics are very, very important. Stomata, trichomes, starch grains, calcium oxalate crystals, those can be analyzed. Microscopes can be of different types: simple, compound, TEM, SEM, for ultra structure and morphology of a particular plant or any particular organelle also. Then TLC chambers, absolutely. You can also go for TLC chambers because pharmacognosy में different phytoconstituents will be present in one particular plant. Curcumin, desmethoxycurcumin, bis-desmethoxycurcumin, etc. Soxylation, absolutely. In soxylation also you can perform, you can extract. Clevenger, again for extraction. Autoclave, sometimes you need to sterilize as well. So for that we can autoclave. Yes or no? Yes or no? Pharmacognosy, again we have certain tests with a specific chemical test for tannin, alkaloid, glycoside, flavonoids, volatile oil resins. You perform some specific test. You can also perform some in vitro culture test. Yes, in vitro culture test we have performed if you're working into natural products. If you're working on cancer, so cancer cell like SCACO2, HeLa2, right? Different cell lines. You're working on Parkinson, any drug, SHSY5I, any cell lines you can perform. Working on heart, H9C2 cell lines. So these are the different herbarium. You can, if you can collect a different set of species with you, drug adulteration if you're checking. Pharmaceutics, absolutely. Zeta sizer, Malvin analyzer is something that you need for zeta potential. Solubility analysis, angle of repose, contact angle, Brookfield viscometer, homogenizer, freeze dryer, batches of freeze dryer, lyophilizers, disintegration test, absolutely. Dissolution, the most important parameter is dissolution. You need to perform a biorelevant media dissolution in a specified biorelevant media. You can also develop bioanalytical method as well, HPLC, for a quantification of the drug release and all. You can perform percent CDR, cumulative drug release. A lot of things that you can perform in pharmaceutics lab. Formulation, granulation, if you're working on novel drug delivery side or conventional. We need certain instruments that for either if it's a tablet, so punching and all, fluidized bed dryer and all. Analysis, of course, UV, IR, NMR, mass, that is compulsory. Besides that, hyphenated techniques like LCMS, atomic absorption, flurimetry, FTIR, all those techniques. You can have X-ray diffraction, right? polarimetry. There are a lot of techniques which you can perform for structural elucidation, for identification, for molecular weight estimation, for conjugation, a lot of techniques. So you can perform either limit tests, you can perform different assays, impurity profiling, spectral analysis, titrations, pharmacoinformatics, mostly tools, NIME, R online, Python, PubChem, Drug Bank, Schrodinger, PDB, Swiss ADM. You need to be well versed with these techniques, softwares specifically. You can perform protein remodeling, pharmacophore remodeling, data mining. You can do QSAR, like quantitative structure activity relationships. Biotechnology, of course, you need a CO2 incubator shaker, fermenter, laminar airflow, very, very important, nano drop to quantify the RNA and all, because many a times when you perform RT-PCR, you need to first quantify the RNA levels. Western blot setup, Biorad, a lot of companies, there are a lot of famous companies like Sigma Aldrich for chemicals. For you, HPLCs, you have Agilent, Water, Shimatsu, you have Biorad system for this. A lot of different companies that are, into setups of these instruments and all. Definitely for ELISA kits, we have ELK, eLab Sciences, ChrysGen, a lot of companies that also deal with the ELISA kits. So that is also another thing that you can opt for. Then you have SDS page, polyacrylamide gel electrophoresis, cell cultures, protein estimation you can perform by Lorry or Biuret or barcodes. A lot of techniques that you can use it. Medical devices, and the medical devices sometimes, chitosan based delivery system. So you need magnetic beads, magnetic stirrer, and all hot plate continuously. Sometimes if you're working on some detail like oximeter or spirometer and all, for that also you can, just find out a lot of instruments. It's still developing, not yet completed. Like if you're working on a patch, if you're working on any particular system, so that all can be developed in a particular lab. So this was it about all the laboratories of the pharmacy that you do, because we have always curiosity ke lab mein aakhir hoga kya, what is in a lab. So I hope that it gave you a clear idea of what exactly you can deal with in a lab. Thank you so much for watching the video. Have a good day, all of you.